Uh, Hello. So, uh, Jubin, you are having any doubt regarding the previous classes?
No, madam. No, madam.
Okay.
Madam, I just wanted to confirm whether this is only a doubt clearing session or it's a usual. Uh, is it like last time, meaning that regular classes?
It is a doubt clearing session. Actually, it is supposed to be a doubt clearing session, but actually, if people are not asking doubt, then like that's why I teach. If someone is asking doubt, I will clear the doubts.
Okay, okay.
Some actually last week someone asked some doubts, so I have all the questions which she asked out. When she joins, I will then tell her. I
Yeah,
don't know whether
okay,
she will
okay.
join or not, today.
Okay, thank you.
So I hope everyone can see my screen. Um, Actually, many people have asked doubts, so I have just added the questions for which they have asked, like different types of questions they have asked, and in mail also some people have asked some doubts, so I have added. So I will discuss the doubts in the end because more people, if like people who asked have joined or not, I have to see. So I hope no one is having any problem with any assignment. If someone is having any problem with any assignment or someone is having any doubt regarding any question or has other kind of question, so they can ask me. Uh, so now it is
Excuse
week.
me, ma'am.
Yeah, yeah.
Ma'am, I have a doubt from week 7 assignment.
Huh,
Maybe week 8, ma'am, previous assignment. In
huh.
this, a question is, in all exceptions, CA is the smaller fragment. And options were like CA, C4, C3, C2.
Can you tell me the question number? It is easier for me to.
Yes, ma'am. Yes, ma'am. Just wait, I will tell you.
Huh.
Ma'am, it is question number 5 from week 7.
Okay, question number 5 weeks. This one that in all except the C.
Yes, ma'am, yes.
Is what doubt you are having?
Ma'am, the answer was C2. But when I saw the videos or when I searched on Google, they were showing that C3 breaks into two, C3A and C3B. And
Huh.
C3 is the smaller fragment. So I want to you explain that why C2 is the answer. Is there any reason or specific thing?
Okay, so here the answer is C2, and you are telling like in the video it is shown that C3 is the like smallest one.
Yes, ma'am. If C3 breaks into C3A and C3B.
Okay. Actually, here the question meaning is in all except the following, C dash A is a smaller fragment. Means in all the complement proteins, the question is which complement protein is an exception. So, like we know that all these complements protein is having like A part and B part, like C2A, C2B, C3A, C3B, C4A, C4B, like
Yes,
this.
ma'am.
So in all the cases, what happens is mostly the A fragment is a smaller fragment and the B fragment is the larger fragment. Correct?
Yes, ma'am.
Huh. So, but in one exception is there in case of C2, the A fragment is the larger fragment, but B fragment is the smaller fragment. So the question is which is an exception in which that particular A fragment is not a smaller fragment. In all except the following means which in which a
Ma'am, but
complement. Ha ha.
ma'am,
Tell.
but in question there is they were not asking about not smaller fragment. They are asking what is the smaller fragment in all except the following. CA is the smaller fragment.
No,
They are not mentioning that not.
in all except means not only, no. In all means in the following
Oh,
options except which one, except you have to put the correct answer will be the exception one in which
okay, ma'am. Okay,
other fragments will be.
there is my mistake.
Maybe in the question,
Um,
like you got confused in this except
yes,
one.
ma'am.
Because
Yes,
in the
ma'am.
other four options, like all the A fragment is a smaller one, except that C2 in which B fragment is the smaller one, but C2A is the larger one. That's why this question is which is an exception, where
Okay,
that
ma'am.
is the smaller fragment.
Got it. Thank you, ma'am.
Uh-huh. So any other question is there?
No, ma'am.
Okay. So now currently week 8 is going on,
Yes,
right?
ma'am.
Huh. So
Yes.
there is no doubt in week 8 also.
Ma'am, I have one, but I am very confused that what will be the answer in first question only.
Okay, but still it is week 8 going on, no? So I cannot currently
Yes,
tell the
ma'am.
answer, huh? So next week you can
Okay,
ask
ma'am.
me if you are having any doubt.
Sure, ma'am.
So today I will, since sir has also started adaptive immunity, I will about adaptive immunity. I have in previous classes already have told about adaptive immunity a lot. So today I will start with this, like how the lymphocyte receptor genes are organized. So, as everyone knows that our uh, T lymphocyte has a high range of diversity compared to the other types of cells, because uh, in our body, like lots of pathogen enters every day, and so this when some pathogen enters, the B cell T cell has to get activated, and all these B cell T cell have some receptors on their surfaces. These receptors should recognize all the pathogens. So you can understand that not all the receptors can recognize the same pathogen or different pathogens. So like there is high diversity among these receptors, where a particular receptor can uh, actually recognize a particular type of pathogen. So from where this diversity comes? So today I will just talk about this how this lymphocyte receptor genes are organized and expressed. And here you can see a microscopic image, where there in the T cells, like in the left diagram, this immunoglobulin heavy chain locus lies in the, as you all know that there are two types of chains in immunoglobulin, or it is like a heavy chain and a light chain. As I have taught in the previous classes also, like there are different types of, there are two types of light chains, kappa and lambda chain, and there are heavy chains. And everyone knows the antibody structure, like it is a Y-shaped structure, and it is having variability, like there is a constant region and there is a variable region. So here you can see, like in this is a T cell and this is a B cell, and you can see that the heavy chain locus is lying in the this area around this rate, whereas in the B cells you can see that is placed in the more and central location within the nuclear matrix. So it depends on the type of lymphocytes and also the organization where this heavy chain, the light chain, how the recombination will take place, everything. So today we are going to learn about how this diversity comes among these receptors. So as I already told, our immune system must recognize a vast array of rapidly evolving microorganisms. As I already told earlier that where we are also evolving every day, the microorganisms also evolves every day. They try to modify their structure to enter into our system. So our immune system must be in par with the microorganisms also. They should also recognize their microorganism, and how they are evolving, our immune system also should evolve. So it should therefore be capable of generating a diverse and flexible repertoire of receptor molecules which can recognize microbial pathogens. However, it has also has to minimize the expression of receptors that bind to self antigen, that is, which is recognizing our self proteins. So, to prevent the different kinds of autoimmune diseases. So, these are the basic requirement the immune system has to fulfill. So, Anjali, hello.
Yes, ma'am. Good evening.
Good evening. So, you had some doubts which you asked me last class.
Yes, ma'am.
Yeah, can you see my screen? Like, these are the following doubts, no, which you asked, like here.
Yes, ma'am. Yes, ma'am.
Yeah, like you had doubt, like totipotent cells can produce, like germline stem cells are example of totipotent stem cells. That was your doubt. But why it is given wrong here? That only.
Yes, ma'am. I asked these questions.
So here the option is germline stem cells that only produce sperms are examples of totipotent stem cells. But can a germline stem cell that only produce sperm can give a whole organism?
Ma'am, I already told you. I applied the logic. I mean, the gametes are formed and then they fertilize and.
Both gametes are needed, to form the zygote and to form a whole cell. But here it has already mentioned that only produce sperms.
Okay. Okay. Yes, ma'am.
Not the ovum part, it is not mentioned here. It is just a trick
Yes, ma'am.
question just to see. If it was like germline stem cells that produce sperms as well as ovum are examples of totipotent stem cells, then you can like understand that yes, they can give rise to a whole organism. But only sperm cells or only ovum cells cannot give rise to a whole organism.
Okay, ma'am, got it.
Another question you had, like, which of the following is not a function of cytokine? Here it is written. You told like it is like it is not a function, not like it is one of the function. You asked this question also, no?
Yes, ma'am.
So here oxygen transport is not a function of cytokine. So what doubt you had?
Ma'am, one of the option was it inhibits cell death, but we have learned that cytokines induce cell death.
Okay. So you are telling like cell death inhibition. Hello?
Ma'am, actually, my question was we have learned that cytokines induce cell death. Ma'am, but
Yes.
why induce cell death and but it does not inhibit cell death. So how can this option? Ma'am, actually, I
Haha,
got
I
confused.
understood. Like you are telling that cytokine induces cell death, and their option is cell death inhibition. Why it is not correct?
Yes, ma'am, exactly.
Okay, okay, I understood your concern. Cytokines actually do cell death. That I also agree. Okay, this question I will mark then because I also think that this 3 and 4 are the. It was a multi-choice question or a single choice question?
Ma'am, it was a single choice question. We have to choose only one option.
Okay. Okay, then I will talk to sir once because cytokine induces apoptosis, so it should be either two or one. Okay, I will ask and let you know again.
Ma'am, actually, if the third option was not given, so we definitely choose oxygen transport. We read that it cytokines directly did not does not involve in oxygen transport, but it
Mm-hmm.
increases the oxygen transport at the site at
Means that
the
increases
location.
the pressure, capillary pressure and all. I
Huh.
understood.
Yes, ma'am. So my question was exactly why we chose this. If the cell death inhibition was not in the option that we then we definitely go for the fourth option. But it was also there and
I understood your concern. Okay, I will mark this question and tell that whether both are correct or not. Okay. And another question you had like this one, that fungi one.
yes, ma'am. Microbiome. Why we
Huh.
can't consider? Yes, ma'am.
So here the question is which among the following statements are or is not correct? it is not question. It is not the question like which is correct. So microbiome present in human body doesn't include fungi is not correct. So what question you had? Like you are confused like whether this is correct or not. The second option.
Ma'am, I had question like if the given option was correct, why we won't include fungi in the microbiome of the human body?
We include, that is why it is not correct. The question is which among the following statements are not correct? Microbiome consists of fungi. So this second option which says that does not include fungi is not correct. So the question is are not correct. Which are the statements which are not correct? So the answer is two only because other two are correct. Like microbes are 10 times more than the number of human cells, and every day an adult human swallow and digest about 1 liter of mucus. So both of these options are correct, but option 2 is not correct. So the answer here will be 2 only.
Okay, okay, I got this. Thank
That
you.
you thought maybe it is like the other way around. It is correct, but ha
Okay, thank you, ma'am.
ha. So anyone is having any other question?
Ma'am, I had one. Actually, ma'am, I want. Sorry for the background noise.
Ha
Ma'am, I wanted to ask for a molecule or any particle to be antigen, is it necessary that it should have all the properties, or only one like molecular weight, or only foreignness, or anyone is sufficient to make it an antigen or an immunogen? Like
uh,
does it have to qualify all the criterias, or any one necessary characteristic is enough to make it an antigen?
no, any one is fine to make it antigen.
Okay, ma'am. Okay, thank you.
Ha. So another person also asked whether this card domain, I don't know whether he or she is present here. So they have asked whether the card domain is why this is oligomerization and not DNA binding. So card domain specifically in RLR uh, helps in the recruitment of the adapters and binding with the card domain of the adapters that help in the downstream signaling process. So here, this DNA binding is not the correct answer, and it is like it helps in oligomerization, where this card domain of the RLR will bind to like the RLR are RIGI and MDA5, which are the two RLRs, and they will have two N-terminal card domain, and it will be helpful in binding with the card domain of other adapter molecules. So it helps in the oligomerization of the complexes and then the downstream signaling. Uh, So this was another question asked, and there are many other questions also. I will discuss after some time. Some other people are joining. I will see because I don't remember the names who asked. So people have asked like they could not understand this table, then this experiments and this diagram, this side effect. So the last I will explain this. So like I was where? Huh. So our immune system needs to identify a lot of pathogens. So it must express diverse types of receptors so that it can recognize different types of microorganisms which is trying to enter into our body. So the antibody structure we all know here. This is a Y-shaped structure, and this is composed of two light chains and two heavy chains. And here, this is the V region, is the variable region, and there are 110 amino acids in the heavy chain and light chain of this V region, and it is highly variable among different antibody molecules. So this is known as the variable region. Here is the variable region, variable heavy chain, variable light chain. So, however, the remainder here you can see CH1, CH2, CH3, Cl. These are the constant chains of both heavy and light chains. These are like constant. Why? Because their sequences are more or less constant in all the antibodies. So, variable region is variable among different antibodies. The sequence is variable so that it can identify a large number of antigens, and the constant region is the sequence is constant in more or less all the antibodies. So the question is, how can a finite amount of genetic information encode a vast number of different antigen binding sites? And how can an immunoglobulin heavy or light chain can be identical to many other heavy or light chains in one part of its sequence but be extraordinarily variable in another? So how in the constant region it is so much similar to other heavy and light chains, but in the variable region, how it can vary so much. So here, uh, first there were two like theories. I will tell. Here are like early sequencing studies indicated that the variable and constant regions of both the immunoglobulin heavy and light chains, they are encoded separately in the germline genome. Germline genome means whatever we have carried from the birth, that our genome, germline encoded genome means whatever was there in our gametes since birth, that is germline genome. And this first it was suggested that our this heavy chain and light chain are like encoded separately from our germline genome. So here it is a molecule of like IgG antibody which consists two heavy chain, two light chain, and it is a variable region and it is a carboxy terminal constant region. And for any given constant region, as I already told, there are millions of variable regions that can associate with it. And for any given variable region, there are only a few constant regions that can be associated with it. So, constant regions will be, a particular constant region will be there, and with that, many types of variable regions can be associated. And in case of one variable region, only a few constant regions can be associated. And the number of different antibody molecules that can be made by a single individual mouse or human is very vast, approximately 10 to the power 13 to 14. Then you can understand every day how, like whenever, not every day, like whenever there is some infection or some pathogen is entering, then how much antibodies are produced in our body, 10 to the power 13 to 10 to the power 14. You can count the zeros and understand this many antibodies are made in our body. But from where this large number is coming? So here you can see the variation in the V region. Like this CH region is constant. CH or Cl region is constant, but the same constant region can be connected to different types of V region, and that will increase the diversity. Whereas here you can see same V region is there, but the constant region is changing. 5 types of antibodies are there: alpha, beta, alpha, not alpha, A, E, M, G, and D. 5 types of antibodies are there. So here you can see these 5 types of antibodies are formed by different C regions. But here the V regions are same. So by this you can, here only in the picture itself you can see 10 different types of antibodies you can count. So like this permutation combination, it gives rise to 10 to the power 13 to 10 to the power 14 types of antibodies. So there were 2 types of theories before the discovery of this recombination among the lymphocytes. First was one germline theory, like each antibody is separately encoded within the germline. Like for IgE, it is having a separate small DNA, and from there where it is getting transcribed. For IgD, it is having a separate DNA from where it is getting transcribed. But however, you can understand whether it is possible or not, because if suppose there are 10 or more antibodies and each antibody is needing 2000 nucleotides to specify both its heavy and light chain, so each antibody would require how many nucleotides? 2 into 10 to the power 10 nucleotides. So you can understand that this much nucleotide is not itself present in our body. Then how they can generate this much antibodies? So this germline theory was discarded. Like not every antibody is encoded by different DNA. Then what is the case? In 1965, 2 scientists, William and Claude,
Excuse
proposed
me,
that.
ma'am.
Huh,
Ma'am, okay, ma'am, it's changed. I thought you are showing some other, because some other slide was visible and you are telling about
okay,
germline
okay,
theory. It is now visible.
okay, sorry. Uh, so this much nucleotides are not present in our body. Our genome is not this much high that every antibody will have different kinds of DNA. So this theory was discarded. So uh, in 1965, 2 scientists, they proposed first that there are 2 types of heavy, like it was already known that heavy and light chains are forming the antibodies, because in the last class I have shown like how the cleaving of these antibodies by different enzymes like papain or pepsin is creating 2 different types of chains, that is heavy and light chains. So they first told that these heavy and light chains are encoded in 2 separate segments in the germline genome. To specify a complete heavy or light chain polypeptide, one of each of the V region and C region encoding segments must be brought together in B cell DNA to form one complete polypeptide encoding gene. So they proposed that there will be separate DNA which is encoding this heavy chain, separate DNA which is encoding the light chain. It is not that separate DNA encoding a separate antibody. It is just each heavy chain is like encoded by a particular DNA, and this light chain is encoded by another DNA. So here you can see they told, like in case of the germline DNA, there is one constant region and many different variable regions are there. Like this is a kind of DNA, and different V's are present in the same stretch with a particular constant region. However, in the B cell DNA, they told like somehow this or many V chains are getting lost in between, and this constant region is joining with a particular V chain. So you can see from the diagram how many V chains are there: 1, 2, 3, 4, 5, 6, 7, 8, 9, and one C is there. So constant is 1. So out of these 9, only one will go and bind with the C chain. Others will get removed. So if like this they are joining, then how many from this diagram itself? One V will join with the one C and form different kinds of 9 molecules. Here 9V with one C will form different 9 molecules. So they can like this they can generate.
Excuse me, ma'am.
Huh.
Ma'am, maybe there is some network issue
Okay.
at your side, or we don't know at our side. Because
Okay,
when you are
my
telling, the slide is not showing, and
okay.
then previous slide was showing. That's why. Sorry for
Here
interruption.
here you can see. Hello. Huh. Now
Ma'am,
you
you are
can.
showing germline DNA and B cell DNA.
Huh.
Yes, ma'am. Now it is visible.
So here you can see in germline DNA, there are 9 different types of variable regions, and there is one constant region. So each of these V can join with C to form a particular VC combination. So from the diagram itself, you can see 9 V can join separately to C to form 9 different molecules. So but it will not form in any kind of cell. It will only occur in the B cell to generate a diverse repertoire of B cell receptors as well as antibodies. So there was some term which is somatic hypermutation theory was given. Somatic means we all know cell, and uh, somatic means to all refers to all the cells of our body except the germline, sperm and egg cells. And somatic hypermutation theory suggests that a mutational process will occur only in the B cells to alter the antibodies in an individual animal and increase the number of total antibodies available to it. So there will be kind of mutation that will occur only in the B cells that can generate a large number of antibodies. And these will not affect the germline genes and would not be passed on to offspring. Whatever antibodies are produced in us in a particular individual, that particular individual will not like transfer this to their offspring. So it is a unique property of a particular individual. And since it is not getting incorporated in the germline cells, so it will not pass to your offspring. So according to this somatic mutation hypothesis, a limited number of germline antibody genes is acted by unknown mutational mechanisms to generate a diverse repertoire only in mature B lymphocytes. So this was given as a like a theory itself to show that how these antibodies are generated. So from a small number of genes only you can get a large number of antibodies. So this was the first experiment done by Nobumichi Hozumi and Susuma Tonegawa, Japanese. And they found this first that multiple gene segments encode the immunoglobulin light chain. And this is that this uh, relocation or this recombination is happening only in the this B cells and not in the germline DNA. What they did, they took some embryonic liver cells. Embryonic liver cells means, you know, that they took from DNA from some embryonic liver cell, which is a source of germline type DNA, and from a B cell tumor cell line, which is a antibody producing cell. So from the B cells, they took some DNA, and from embryonic liver cell, which is a example of a germline DNA, because in embryonic state and liver also this divide into, like, the cells in the liver divides. So from the embryonic liver cells, they took the DNA, and from the antibody producing tumor cell, from the same strain of mouse, they took the uh, DNA, and they digested with some restriction enzyme. Restriction enzymes are certain enzymes which will cleave after a certain sequence in the DNA. Like they will act only on the certain sequences in our in any DNA, and will cleave that particular portion. So they cleaved this DNA with some restriction enzyme, BAM each one. Then they separated these DNA fragments by a gel electrophoresis, and I hope everyone knows what is gel electrophoresis. This is a technique in which we separate DNA. We can separate DNA or through SDS page, or we can separate the proteins, different kinds of proteins based on their molecular weight. And they found the after separation of this DNA, after cleaving this DNA, and with agarose, we use agarose for DNA gel, and for protein we use SDS. So here they have ran agarose gel and have found uh, they found out the location of the fragments containing V and C regions with specific probes for the entire K light chain and for the C region alone. So here they after that, after running the gel, they have done a southern blot. So southern blot is for where we use probes against some DNA to visualize it in a membrane. In case of western blot, we do it with some antibody which will bind to a specific protein. In northern blot, again we will do for RNA, where some probe will bind to the uh, RNA, and we can visualize in a membrane. So here they have used some probes which they know was specific for the kappa light chain. So there are two types of light chains. One is lambda light chain, another is kappa light chain. So here they have done these experiments for the kappa light chain. So with the kappa light chain, they have designed some specific probe and have probed it on the membrane. And for the V and C region only, they have made the probe, that is the constant region and the variable region, and also for the C region alone. So here after that, they found that in the embryonic liver, the DNA sequences encoding the V and the C region were located on different restriction endonuclease fragment. Means when a big chunk of DNA is there, suppose 1 kb of DNA is there, they have cut it into small pieces with BAM1 restriction enzyme. So it will form many pieces, maybe it will form 100 pieces. So in that 100 pieces, when they probed it with this probe against this uh, V and C region, they found that this DNA sequences were located on different restriction endonuclease fragments. Means it is not located in the same one. The C region here you can see is present here. This light chain means V and C region is present here. But however, in the B cell, they saw that the two sequences are co-located on a single restriction fragment. Means the V and
Excuse
C
me, ma'am.
region. Hello.
Ma'am, which slide you are showing? Ma'am, it is
Okay.
lagging very much. I don't know why.
Okay. I don't know
One
whether
message also.
uh, it is 9.
Yes,
Slide number
ma'am.
9.
Okay, ma'am.
Ah. It is visible, no?
Yes, ma'am. Now it is visible. When I say to you, it comes and then I don't
Okay.
know what is happening.
Okay. So as I already told, in this picture you can see, suppose in germline DNA, they are cutting with BAM H1. So it is cutting uh, here, here, then different fragments it is cutting. So here you can see this blue colored V region is far away from the C region. So when you are cutting the DNA with the restriction enzyme, it will give separate region and it will give many fragments. But in the B cell DNA, what happens? These two becomes closer. This V is coming close to C. So when it is cutting the restriction fragment, number one, restriction fragments will also be less in number compared to this germline DNA. Number two, this cannot cut in between this V and C. So it will appear as a single fragment. So when you are probing this with V and C region, it will probe in a single type. But in germline DNA, when you are probing this band will appear separate, this band will appear separate. So you are probing the germline DNA, you can see one band. That band is with the C region, probe is binding with this C portion, this C portion. So here you can see as usual one band. But here when you are probing this with V as well as C region, it is also binding with V region is here, C region is here. So two bands they are giving, two separate size bands. But in case where there is a joining of this V and C region, this probe is binding here, this probe is binding here. So when you are probing with C region alone, this C region is binding here, no problem. So it is giving one band. And when you are probing with both the V and C, this V and C also binds to this region, and in the same region it is giving a band. That means something has occurred in the DNA which has this V and C region to come closely. This gave the first idea to this scientist that there are some mechanism which is occurring and there are multiple gene segments that can encode this immunoglobulin light chain. So, and this is only occurring in the antibody producing cells and not in the germline DNA. So everyone understood this experiment. I know it's complicated a bit. I hope everyone understood, because actually these are the basic experiments which were done initially to prove some basic points which we are learning today. So that's why these experiments are kind of interesting. If someone wants to discover something new, they can get idea from these experiments when there was no much technology available there in the 90s, 80s. They have actually proven these facts, like how these things are occurring inside our body. So, so the antibody K light chain is composed of three family of DNA segments. They found out there are variable and constant region, and there are one J or joining region in between also. So uh, one single C segment, as I already shown, is located 2.5 kV downstream from the J segment, and there are three. Here you can see three different types of V segments. You can see VK, three different types of VK which are present upstream of this J segment. However, during B cell development, when this B cell is developing, DNA recombination will join one V segment and one J segment in each B cell. So here one V is joining to one J which is joining to the C region. So like this there can be permutation combination between this region and it can generate a different types of K light chain. In case of heavy chain, apart from V, J and C, there is one region which is a D region. So there will be a recombination between this V, D and J to form the variable region of the heavy chain, immunoglobulin heavy chain. So D stands for the diversity region. And here recombination between three gene segments, the V, D and J segments is required to encode the variable region of the heavy chain of the Ig molecule. And the D region and VD and DJ regions encode the regions of greatest sequence diversity in the antibody heavy chain. So here you can see so many D regions are there, so many J regions are there, so many V regions are there. So any V can join with any D can join with any J, and there will be a large number of variation if all these segments are mixing and matching among themselves to produce different types of antibody molecules. So here you can see this V is joining with D, one of the D, one V is joining one of the D, which is joining with one of the J, and this is a C mu, means the constant region. Mu means it is a IgM antibody. So it is forming a particular IgM antibody. So here multi-gene organization of immunoglobulin genes, you can see in mouse how they are organized or they are recombined. Here this is the light chain with V1, V2, V2, Vn it is written. We also don't know what are the number. Vn means it can go till infinity, V to the Vn. Then there is J, there is C segment. And here this is a again another light chain. There will be two light chain, kappa as well as lambda, and this is a heavy chain DNA in which the K, lambda and H chain gene segments. This heavy chain will have, like here also you can see this VH to the VHN, then D, here you can see D till 14, then here J 4 J segments are present, and C, depending on the type of the immunoglobulin, it is different types of C's are also present. So here the kappa, lambda and H gene segments in the mouse genome are illustrated in such a way so as to indicate both the large number of V gene segments and the overall size of each of the V gene loci. So here you can see this is just a diagram, but here they have presented a overall number of V segments. You cannot even count this, how many are there. This all the like the dashes. Here also same, this is the light chain kappa, this is the light chain lambda, and this is the heavy chain. So this many number of variable regions are present which can go and bind to any J, any D and will form a large number of antibodies. So you can see the combinatorial antibody diversity in humans. For example, estimated numbers are given. Still now no one is sure, but somehow estimated numbers are given how many V regions, how many D regions and how many J regions are present. Here you can see nature of segment V. Estimated is 45 are present. 45 such sequences are present. And number of K chain segments 41. Number of lambda chain segments 33. Then this D 23. Then J 6, 5, and 5. They come in case of light chain. D is only present in heavy chain, not present in this light chain. So you can calculate if this B is joining with D and this D is joining with J, then 45 into 23 into 6. Possible number of combinations is 45 into 23 into 6, 6210. Here also, if this V is joining with this 41V, either of them are joining, then 205. Then 33 into 5, this lambda chain, then it will be 165. Then you can understand how many combinations are possible since so many are there. And obviously with different types of heavy chain can go and bind with different light chain, and then it will, if you multiply this with this, then this with this, then it can give more numbers. So this many types of antibodies can be formed in our body. So in case of lambda light chain, in mouse they have seen that it has undergone a deletion event such that there are fewer V than V gene segments. However, than their K chain counterparts, mouse lambda chains are less significantly less diverse. And in mice, 5% of the light chains are of the lambda isotype, whereas in our body, 40% of the light chains carry the lambda constant region. And recombination between V and J segments can occur only between V sequences that lie upstream of their J partners. So here you can see this V region is located upstream of the J partners, so recombination can occur there. So, and heavy chain, as I already told, it consists of V, D, J, and C, which encode the variable region of the heavy chain, and they are located upstream of a set of 8 constant region gene segment. There are 8 constant regions in the heavy chain, and in humans and in mice, it differed between strains and in different individuals. This V, D, and J. It did not be that whatever estimated numbers it is written here is present in all human beings. Someone will have more, someone will have less. And the human V locus has a similar arrangement with approximately 45 functional V segment, 23 functional D segment, and 6 functional J segments. They are transcribed in the same direction as the constant region gene segment. Obviously, it will be transcribed in the same direction as the constant region, because otherwise, if there is a opposite transcription, the whole antibody molecule will be opposite. Like the V region will come down, the FC region will come above. So it should be in a directional manner. Recombination among the various V region gene segments generate a diverse repertoire of antibody containing sites. So uh, here you can see, like VDJ recombination in lymphocyte is a very highly regulated sequential process. It does not occur randomly, and first V should join to D, then VD should join to J, then VDJ then will join to the constant region. And there are different stages through which you can mark whether a B cell is a mature one or it is an immature cell. For example, here you can see pre-pro B. Pre-pro B means these are the first stage, this B cell, and it is having no receptor on its surface. And these are the different kinds of proteins or the molecules which are involved in the which are present on the surface of these B cells. And this RAG1 is the recombinase which helps in this recombination process, and this proteins, and the CD proteins will be present on the surface of these B cells. So then after the pre-pro B cell, there will be uh, early pro B gene, early pro B cells, where there will be joining of D and J. So here also no immunoglobulin receptor is still formed. Here you can see this Ig alpha beta co-receptors are formed. Then late pro B. Here the DJ is joining with V variable region and it is forming VDJ. Here also this is first the heavy chain rearrangement is occurring. Here all these markers are present. Then first checkpoint is it is forming a large pre-B cell. Large pre-B cell, it is known as pre-BCR. So now you can call VDJ is present. So pre one immunoglobulin or pre-BCR or B cell receptor is formed. Here after that it is forming small pre-B cells. Like the heavy chain has joined VDJ, but now rearrangement with the light chain occurs. So there is decreasing level of pre-BCR and increasing level of the small pre-BCR. And after that, this recombination form, this recombination is over and there is a complete uh, expression of this receptor, and here you can see it is forming a IgM antibody here. IgM antibody, but still it is immature. It has not re, uh, like recognized its antigen, and thus it to become a mature B cell, you have to first recognize the antigen and become active, and then clonally you should proliferate and differentiate and form more and more B cells. So this is the process by which this sequence should be there, not like randomly any V is joining J, any D is joining J, but first there should be joining of the D segment with the J segment, then this DJ segment is joining with the V segment, and then after that there is incorporation of the light chain segments. Then only it is forming a complete receptor on the surface of this B lymphocytes. Here you can see, as I already told, the pre-BCR. This is a pre-BCR which is having this VDJ here. This is the heavy chain, light chain joining still happening, and these are the different co-receptor molecules as I already told, signaling components which is required, and here you can see the mature one where the light chain joining has almost finished. All the co-receptors are in place and now it is ready to become a active receptor or a active antibody. So how this recombination occurs? So this recombination occurs by some specific sequences or what is known as the signal sequences. So there are recombination signal sequences which are present on the DNA. On this DNA, you can find this sequences which contains conserved heptamer and a conserved AT-rich nonamer. Heptamer means seven nucleotides will be there. This 7 sequence you can see is heptamer, and nonamer means 9 nucleotides will be there, and they are separated by these 23 base pairs. Here also same. So these are the recombination signal sequences. So it consists of a heptamer, here CACA-GTG, and the spacer 23 base pair or 12 base pair, and the second conserved nonamer AC, then 5A, then double C here. So these two types of recombination signal sequences have characteristic locations within lambda chain, kappa chain, heavy chain germline DNA. During DNA rearrangements of the Ig heavy chain and light chain, the gene segment adjacent to the 12 base pair RSS can join only with the segments adjoined to the 23 base pair RSS. So here K chain, you can see this spacer 12 chain 12 base pair 23 base pair. Here also similar lambda chain, and here also heavy chain light chain. Here you can see. So only this DNA segment which is adjacent to this 12 base pair RSS can join with this segment which is adjacent to the 23 base pair RSS. So you can understand that this is occurring in a sequential or in a directional manner, not like the other way down. Otherwise, the sequence of the antibody or the receptor will be reversed. So this recombination between the gene segments is required. Here you can see this is a germline light chain DNA. This is germline heavy chain DNA. Here the V cluster is there 1, 2, 3. J cluster 1, 2, 3, 4. This is a 12 base pair spacer and this is a 23 base pair spacer. And this is a constant region. So this is recombining with each other. And these are mediated by different kinds of proteins. As I already told, one protein is RAG. Some recombinase is there which is mediating this recombination, and you can see this middle region is removed from here. So segment 1 and 2 of J is removed and this V3 is joining with J3 here. V3 is joining with J3 here, and this V1 and V2 is written, then J4 is also retained, and this is forming a recombined VJ gene in B cell. In case of heavy chain DNA, also here you can see this D cluster is having 12 to 14 segments and J cluster is having 4 functional segments. There will be recombination between these two, and the segment in between is removed, and here they are joining DJ arrangement, and then one V will again join with DJ and it will form a VDJ. It is joining and forming a recombined VDJ gene in B cell. So here is the exact mechanism. So everyone can understand what I am speaking, or there is some issue or doubt is there. Okay, I hope everyone understands. So So here is just a diagram which shows how this recombination is take place. As I told, RAG1 or 2 the proteins, and these two proteins are binding to this recombination signal sequence. Here one recombination signal sequence, you can see two are there. It is having some 12 spacer and 23 base pair spacer, and they are helping in formation of a synapse kind of thing between a V and a J section. This protein is allowing this region to come together, like this portion is going this way and these two portions is coming. There will be single stranded break at the exact 5' border of the RSS bordering both the V and J segments. Here you can see there is a break which is occurring here, and there will be a hydroxyl group is liberated, and at the 3' end of the coding strand attacks the phosphate group on the non-coding strand. Here you can see a phosphate group is there. This OH group, this is attacking this phosphate group, and there will be a hairpin here. For hairpin like structure it will be formed, and there uh, this signal and joining ligates the ends of the two RSS septameric sequences that were originally contact with the V and J coding sequences. Here you can see this monomer is there, 12 base pair, 23 base pair, and sequence at the signal junction results from the joining of the two heptameric regions, and it is this signals is getting discarded here, and there is opening of the hairpin that can lead to a overhang, a 5' overhang or a 3' overhang, and then there can be different types of Artemis means different kinds of defects also, which is I planted also 3' overhang or 5' overhang. Then there will be cleavage here to generate like in the cleavage in the hairpin there is some sequence, and there is cleavage of this uh, hairpin that will generate a this nucleotide. Here it is cleaved, so TCGA it is opening. Then there will be uh, there will be DNA repair enzymes which will fill up this gap, and there will be ligation of this V and the J chain. Here you can see V or J chain is getting separated here, so there will be ligation here. So after the ligation is done, there will be this. This is in case of the light chains, it will light chains. This V and J is joining. So, but in case of heavy chains, there is still this joining of this D and J first, then the VDJ. So first here also similar like the D, there will be cleavage and there will be formation of these complementary sequences which will be joined by the DNA repair enzymes, and this DJ will join. After that, similarly, that VDJ also will join like in the light chain. So this is just an overall view how these enzymes are coming, breaking the bonds, ligating, repairing, and forming the like how they are recombining the VDJ with one another. So, what are the 5 mechanisms that we learned about that generate antibody diversity? First is that multiple gene segments exist at heavy and light chain loci like VDJ and VJ. These can be combined with one another to provide extensive combinatorial diversity. Then heavy chain or light chain diversity. Same heavy chain can combine with different light chains and vice versa. Like same light chain can combine with different heavy chains. Then this combination of different heavy and light chain pairs to form a complete antibody molecule, which provides further opportunities for increasing the number of available antibody combining sites. Then this P nucleotide formation, which I showed actually. Here some P nucleotide formation is happening. So this results when the hairpin, DNA hairpin at the coding joint of heavy and light chain is cleaved asymmetrically, and then filling in the single-stranded DNA piece resulting from this asymmetric cleavage generates a short palindromic sequence. P nucleotide results in the formation of palindromic sequence, that is, which can be read in the, here you can see, like it is forming a palindrome means the both the sequences will be similar from both the ends. Like AGCT is there, here also AGCT. TA. So it is forming a palindromic sequence here, which is necessary for this generation of this uh, VDJ and or VJ or like DJ or VJ. So exonucleus will trim sometimes, and VDJ and VJ junctions, which can cause loss of the nucleotides. And there will be nucleotide addition by some nucleotidal transferase in heavy chain B, VD, and DJ junctions, and from DNA polymerase mu in both heavy and light chain. So these are the different kinds of antibody like mechanism by which naive B cells can generate different antibodies, and there will be diverse antibodies which will be formed. However, each B cell should express a specific type of receptor. It is not like each B cell can form a different types of receptors. Otherwise, one B cell will recognize different types of antigens, but that is not this that is not the case. Like one B cell will differentiate to form a clone of that particular B cell. It is not like different types of B cell receptor will be formed. It will be produced in one B cell. So there are four alleles, as we know. In one gene contains of two types of alleles. So here, if we consider heavy chain as well as light chain, there will be total four alleles that will encode both heavy chain immunoglobulin receptor genes and their K light chain partners. If all the four alleles were undergoing rearrangement, then the host B cell can express up to four different antigen binding sites, with each heavy chain binding to each K light chain to create a new receptor type. So if all the four alleles of two of heavy chain and two of light chains are combining with one another, then they will form different types of receptors. And if both K and lambda also are, then rearrangement is occurring between K and lambda also light chain, then more antigen binding sites could be expressed on each B cell surface. But however, this is sounding like it will generate more and more number of receptors, but the problem is the more and more presence of more than one receptor in B cell, it will make it very difficult for the organism to prevent the expression of receptors that recognize self antigens. If any random things are joining and any random receptors are coming up, and one B chain with more than one receptor are coming up, then it can lead to the recognition of self antigens, and that can lead to autoimmune diseases. So there is a mechanism which is known as allelic exclusion, in which the organisms have developed a mechanism by which B cells ensure that only one heavy chain allele and one light chain allele are transcribed and translated. So not all the four alleles will be recombined and joined and form different types of receptors, but in one particular cell, only one allele of heavy chain will recombine with one light chain allele, so that there will be formation of only one type of receptor on the B cells. So this is to reduce the number of receptors in a particular type of B cells. So receptor editing of potentially autoreactive receptors occur in light chains. So here you can see this is a germline DNA. So first rearrangement is occurring between V3 and J3. Here V3 is joining with J3 and all this J1, 2 are discarded. But it is resulting in a light chain with antibody heavy chain, which is resulting in a formation of an antibody which is autoreactive or autoimmune. So what the cells will do, they will again undergo another recombination event. Here this 3 and 3 has already joined, but they will remove this 3 and this V3 and J3, so that this receptor should not be autoreactive, and there will be second rearrangement between V2 and J4, in which they will join together and form in a non-autoimmune antibody. So Our cell knows what is harmful for us and what is beneficial for us. So it will try to eliminate as much as possible all the receptors which can be autoreactive and which can cause autoimmune disease. So in the event of a productive light chain rearrangement that leads to the formation of an autoreactive antibody, and additional rounds of rearrangement may occur between upstream V and downstream J segments. Here the primary rearrangement between V3 and J3 is edited out with a secondary rearrangement between V2 and J4. So not only one round of rearrangement will occur, but multiple rounds of rearrangement can occur till the cell is convinced that this is a healthy type of cell and not against any kind of self antigen or any kind. It is only recognizing a pathogen which is coming from outside. So as I already told, a differential expression of secreted and membrane-bound forms of immunoglobulin mu and delta chain is regulated by alternative RNA processing. So here you can see as you all know, two types of B cell receptors will be there. When we are calling a BCR, it will be membrane bound, and when we are calling an antibody, it will be a secreted one. So depending on that, this like whether we need B cell receptor which will act as a APC or antigen presenting cell to see T cell, or whether we will require an antibody which will be secreted, this VDJ recombination will take place. For example, here you can see the polysite and the constant exons here. This is a IgM. C mu means IgM. This C delta means IgD. So first what happens? There will be polyadenylation and there will be recombination, like polyadenylation of the primary transcript there are two polyadenylation sites here. Poly site here after this constant region delta 1, delta 2, delta 3, and poly site here at constant region mu 1, mu 2, mu 3. So if there is a polyadenylation in this region, then it will form a IgD. If there is a polyadenylation in this site, it will forming a IgM. So here, since it is having a polyadenylation here, it is forming a IgD, and if it is happening here, it is forming a IgM. Here similarly, if there is this is a site, there are different types of polyA1, polyasite 1, polyasite 2, polyasite 3, polyasite 4. Here you can see two green regions M1 and M2. These are mainly for the membrane B cell receptor, and this is for the if suppose polyadenylation is occurring in M2 here, that means this whole transcript will be considered and it will form a membrane bound antibody, that is a B cell receptor. But suppose polyasite this polyadenylation is occurring, then it will form a secreted mu chain which is a antibody. So depending on this poly signal, it will decide what type of antibody it will form, whether it will be IgA, IgM, IgE, IgD, or whether it will be a membrane bound antibody or whether it will be a secreted antibody. Here also you can see this this is the secreted as well as the membrane bound. Here is extra hydrophobic segment is present which is responsible for uh, embedding of the receptor on the surface of the B cell, and here this M membrane or hydrophobic segment is not present in the antibody, secreted antibody form. So this membrane form, the hydrophobic sequence is coming from this M1 and M2 segment. So you can understand how much our cell are actually uh, what to say, trained to form different kinds of molecules and depending on the need of the hour. So a mature B cells simultaneously can express membrane-bound IgM and membrane-bound IgD receptor that bear the same binding site. The variable and constant regions of these two heavy chains classes are synthesized on one long pre-mRNA, and differential RNA splicing is used to generate membrane forms of mu and delta heavy chains. As I already told, IgM and IgD are on the same DNA segment, so it can both one particular B cell can express first both IgM and IgD receptors, but however, after that there will be RNA splicing that can generate this either mu chain or the delta heavy chains. Following antigenic stimulation, the cells differentiating into antibody secreting plasma cells that generate secreted forms of IgM or IgD, and shift from synthesizing membrane-bound to secreted IgM is controlled at the level of RNA splicing and is one of many changes that occur during B cell differentiation into plasma cells. And the expression of the constant regions of heavy chain classes other than mu or delta requires a DNA recombination event in which upstream constant region or regions are deleted. This event is termed as class switch recombination. So this is since this mu or delta is or the IgM or IgD is synthesized from the same fragment, but other classes of antibody are uh, synthesized by a mechanism known as class switching. So in the next class we will learn what is class switching and how, like, apart from IgM and IgD, other uh, antibodies are formed. So till now that is all for today, and next class we will learn about the T cells also, because today it was all about antibodies and B cell receptors. So anyone has any doubt the class or anything they couldn't understand? Okay. So, so here I will discuss this doubts which people had. For example, some person asked that she is having a difficulty in understanding this table. So here, this is a table where these are the different kinds of TLRs, TLR 2, 4 and 5, and this is having polymorphism, which is all this is having polymorphism, which is uh, connected with some disease. For example, here this thymine in the position 597 is changed to cytosine, and here arginine is changed to this uh, glutamine, and FAB region of antibody comprises both variable and constant region of both heavy and light. Uh, Hello. Uh, FAB region means the antigen binding region. So antigen binding region is consisting of the heavy chain as well as the light chain. Only the variable region, not the constant region. Like FAB portion is the variable region. I will write in message. This is the FAB region as I show. This is the FAB region where the antigen is binding. So this is VHCH, VHCH, VLCL, VLCL. So it is consisting one of the constant region and all the variable regions of both the heavy and light chain. So I was where this TLR2, this is having some mutations or polymorphism where this tyrosine is changed, sorry, not tyrosine, thymine is changed to cytosine, and this arginine is changed to glutamine, and this is when this type of mutation occurs, there is a increase in the protective effect against leprosy and predisposition to tuberculosis, or there can be any staphylococcus infection. And in TLR4, there can be change in the from this amino acid like this aspartic acid to glutamine, and this uh, thymidine to inosine, which can actually lead to systemic inflammatory syndrome and septic shock, decreased risk of atherosclerosis, no association with susceptibility or severity of rheumatoid arthritis. And in TLR5, a dominant negative stop codon mutation can happen, which can increase the susceptibility of the individual to pneumonia caused by Legionella pneumoniae and negatively correlated with Crohn's disease. So, this table mainly uh, tells about the different kinds of polymorphism that can occur in different individuals, which can lead to the susceptibility of that individual to different kinds of disease. So another is someone has told whether when the irradiated mice with were not infected die in the transferred experiment. Why? They received the immunocompetent cells from the syngenic ones, so couldn't those cells remain stored in the irradiated mice and restore their HSC deficiency? What is the exact reason of their death? I could not understand much well what he or she wants to tell, but maybe uh, in this experiment, transfer experiment, it is to understand whether a particular cell type is playing any crucial role in infection or not. So what they have experimented is, first they have infected a group of syngenic mice with virus. The other two groups they have irradiated with the any kind of UV or X-ray. And what they have done is, uh, first uh, they have taken two types of mice. One is they have infected with the virus, and another is they have not infected with the virus. And from there, they have taken the hematopoietic stem cells from both the cases, from without infection with infection. And here they have irradiated with these two groups of mice, and they have transferred that hematopoietic stem cell after infection to these groups of mice and who were irradiated, and another from with the mice they have not infected, they have taken the hematopoietic stem cells. So in case of the mice in where they have infected and taken the cells, the hematopoietic stem cells have differentiated, and but in case of the uh, person or the mice where the it is without infection, they have taken the cells without from the mice without infection and have put it into this mice, then irradiated, then they found that they did not survive, they died. Why? Because the hematopoietic stem cells could not differentiate in that in which no infection was there. Since in case where there is no infection, the hematopoietic stem cells won't divide into different kinds of myeloid as well as lymphoid progenitor cells. Why? Because they will because they have not got stimulated with any kind of antigen, and they cannot uh, differentiate into different cell types, and so that irradiated mice died. But in case of that, it didn't die because they have formed different kinds of cells irradiated after infection, which survived this after infection, in which they have taken the cells and have transferred into the other mice which was irradiated, they survived. So the since the hematopoietic stem cells were not active in case of the mice which was not infected, the these mice died where the cells were transferred. Another one was someone has asked what is happening in this video. This is just a signaling uh, which sir has taught, signaling of various TLRs and PRRs. This is just a signaling pathway by different kinds of TLRs, different kinds of NLRs, and different kinds of RLRs, which they have he has taught. And this is a paper through which she or he has asked for providing the research paper. So here you can see different kinds of TLRs are there. Some are membrane bound or cytoplasmic TLR, some are endosomal TLRs, and they are activating this different kinds of adapters like MyD80, TRIP, etc. They are all are converging in the same pathway, like they're activating the IRF7 complex or the IKK complex, which is activating NF kappa B, and it is activating the type 1 interferon pathway, which is leading to the production of the pro-inflammatory cytokines. Here also different node-like receptors are there, which is activating it via this NF kappa B pathway. And here also, here you can see some receptors are there. Here RLRs are there, which are also activating this NF-kappa B pathway, which is secreting this pro-inflammatory cytokine. So all these are converging in the same pathway here. Just the initial molecules are different, which is recognizing different types of pathogens and their molecules. So I will provide the paper to the person who has messaged it, mailed. Then another person has mailed like why there is a low WBC count due to interferon therapy, but earlier you had told that interferons are responsible for antiviral state and activation of macrophages. Please clear this contradictory doubt. So, side effects of interferon therapy has shown that the interferon therapy need not always, like whenever there is something excess in our body, then it is not good for us. And it has shown that the interferon sometimes affect the bone marrow and there is a suppression of the bone marrow by the interferons. So there can be like sometimes people who are given interferon therapy, it is shown that there can be decrease in the blood cell count due to the suppression of this bone marrow or suppression of the differentiation of the HSCs, which can occur due to excess interferon therapy, and that can lead to the low white blood cell count, which can actually increase the risk of infection. So, it is true that interferons are responsible for antiviral state and activation of macrophages, but sometimes in patients it has been seen that it can also regulate the like the overexpressive activation of the immune system. It can regulate and there is decrease in the white blood cell counts, or there can be less thrombocytopenia, destruction of platelets, or it can also cause anemia. So these are the proven facts which are seen in the people. uh, Activation of macrophage and antiviral state is completely true for interference, but there can be side effects also, which includes low white blood cells counts and or anemia and other like thinning hair, skin rashes, flu-like symptoms, etc. So, anyone is having any more question? So that's all today for today. I have explained, or if someone has not understood anything, please you can drop a message in the portal, or you can ask me also here in the message or in the next class. And Anjali, your doubt I will clear tomorrow. Not tomorrow. This Tuesday, next Tuesday, I will clarify with sir and ask you whether what that regarding that cytokine function, whether it is correct or not. Thank you everyone. So this is all for today.